We all know that heart has its own pacemaker, the sinoatrial node or SA node, which generates the impulse, and the heart beats as a result. On top of it, there are other factors which control the cardiac function. Okay, so you have autonomic nervous system, sympathetic and parasympathetic branch. Sympathetic branch will increase the heart rate and also the force of contraction. Parasympathetic, on the other hand, will decrease the heart rate. Okay. What else? So the cholinergic receptors, so the parasympathetic nerves, they work through cholinergic receptors, and these are muscarinic type of receptors which are present in the cardiac tissue. You know the other type of cholinergic receptors? There are two types from your autonomic nervous system knowledge. There are two types of cholinergic receptors. Cholinergic because it's acetylcholine that binds to these receptors, which is released at the parasympathetic nerve endings. Remember the two types of receptors? Muscarinic and anyone? Nicotinic. Okay, so muscarinic receptors and nicotinic receptors. In the heart, you have muscarinic receptors. Okay, acetylcholine will bind to the receptors and will perform its action. Now, adrenergic receptors, which are present in the sympathetic nervous system, these receptors are two types, alpha and beta. In the cardiac tissue, we have beta receptors. Okay, beta adrenergic receptors, and the two ligands that bind to adrenergic receptors are noradrenaline and adrenaline. Okay, all right. Does anyone know these neurotransmitters will be released when there is autonomic outflow? So when there is sympathetic outflow, the sympathetic nerves will release acetylcholine, right? It will bind to muscarinic receptor. When there is sympathetic outflow, sympathetic nerves will release adrenaline, and it will bind to the adrenergic receptors. Do you know any other source of adrenaline in the body? What is that? Anyone? In addition to sympathetic nerve fibers, where else adrenaline or noradrenaline can come from? Adrenal glands, right? Adrenal glands. Okay. Yep. So that's why when you have, this fight or flight response, you have a lot of adrenaline released into your system, your heart rate goes crazy. Happens, right? Yeah. Okay. All right. Myocardial cells, all right, similar to skeletal muscle cells in appearance. They have sarcomeres, intercalated discs. So this is something which you find in the myocardium. These cells are branching and networking. Skeletal muscle, each fiber is separate on its own. There is no branching and communication between the fibers. In cardiac muscle fibers, these fibers branch and they communicate to each other. The benefit of this is when you stimulate one cardiac muscle fiber, the stimulus will spread to other muscle fibers. So the whole heart or the whole ventricular chamber will contract as a single unit, rather than contraction traveling as a wave across the wall of ventricle. Okay, so because of this communication between cardiac muscles, if you stimulate one cardiac muscle, the signal will spread to adjacent cardiac muscles. Okay. You all know the actin myosin proteins, which are the contractile proteins present in the muscle, which are present in cardiac muscles as well. And myocardium requires a lot of oxygen because the muscles are contracting, relaxing all the time. So the oxygen demand is pretty high. Okay. Myosin actin, these proteins are involved in contraction, cross-bridging. You remember this? I'm just mentioning these things here, assuming that you already know these stuff, right? So you have actin and myosin filaments. They slide upon each other, and the muscle shortens during contraction, and it lengthens during relaxation. Okay, movement of these actin myosin along each other. There are different types of calcium channels which are present on the myocardial cells. Okay, L type and T type. There is troponin, tropomyosin, which are also proteins associated with myosin, where calcium will bind, ATP will be used, and muscle contraction will occur. Right? Do these names ring some bells? Yeah. Anyone online? Do these names ring? So they ring some bells. Okay, that's good. Right. Okay. So they sound familiar. Perfect. Okay. Now, let's look at another concept now, which is called cardiac output. The main purpose of heart is to act as a pump. So the amount of blood that heart pumps per minute is called cardiac output. How can you calculate this? If you know the amount heart pumps with every cardiac cycle or every heartbeat, and if you know how many times heart beats in a minute, you multiply the two, you get cardiac output. Yeah? Okay, so heart rate multiplied by stroke volume. Stroke volume is the amount of blood that the ventricle will pump out when it contracts every time. The average value, for example, of heart rate is 70 beats per minute, and the average stroke volume in an adult is about 80 milliliters. So every time a ventricle contracts, 80 ml of blood will be pumped into aorta in case of left ventricle, and 80 ml will be pumped into pulmonary artery in case of right ventricle. Okay? So when you multiply the two, the cardiac output is about 5.6 liters per minute. The amount of blood heart pumps in a minute. Easy? Okay. Now this is a very important indicator in heart failure. The cardiac output actually drops. So the heart is unable to maintain cardiac output and the heart fails. Okay. We'll talk about heart failure next week. Okay. Now, stroke volume, just to show you in a more graphical manner. So this is left ventricle, and this is relaxed. So it is filling up with blood. This phase is called diastole. Yeah. So heart is filling up, and now after filling up, it contracts, and the blood is pumped out into aorta. Now, usually the left ventricle at the end of diastole has about 120 milliliters of blood. And this is known as end diastolic volume, or EDV, end diastolic volume. When it pumps, 80 ml leaves the ventricle, still 40 ml remains behind. So it can't empty completely. Some amount of blood is left behind. This is known as end systolic volume. Okay, end systolic volume. So if I want to put a formula there for stroke volume, that would be stroke volume equals end diastolic volume minus end systolic volume, right? That will give you the stroke volume, which is about 80 ml in adults. Make sense? Okay. Now, if you want to change cardiac output, right? If you want to change cardiac output, you can do it by two ways. Either by increasing stroke volume or by increasing heart rate. So if someone's heart rate goes up, for example, let's go back to the previous picture, previous slide. So stroke volume remains 80, for example, and this person's heart rate, for example, becomes 100 per minute. From 70, it becomes 100. Stroke volume is unchanged. What's the cardiac output now? 80 times 100? It's 8 liters per minute. From 5.6 liters, it becomes 8 liters per minute. So you can imagine by changing the heart rate by a few beats per minute, you can alter cardiac output to a large extent. Then if you increase stroke volume as well, together with heart rate, you will have even higher increase in cardiac output. Okay? All right. So the factors that control heart rate, let's look at those factors first. What can increase or decrease heart rate? We saw earlier, sympathetic nervous system can increase heart rate. Parasympathetic nervous system can decrease heart rate. Okay? Hormones and ions. Circulating catecholamines, so the ones that are coming from adrenal gland, will also increase heart rate. Okay? Thyroid hormones, which have a general metabolic effect, also increase heart rate. Decreased potassium in the blood will also increase heart rate. What else? With age, okay? With exercise, with higher temperature, again, heart rate will change. So these are some of the factors that could determine heart rate. Now, how does sympathetic and parasympathetic system act on the heart? Sympathetic fibers come from the sympathetic chain, and as you can see, these fibers, they are supplying the ventricle, the atria, and also the SA node, right? When you look at the vagus nerve is sympathetic or parasympathetic? It's parasympathetic, right? Parasympathetic, you see the fibers here, they are not supplying the whole heart, right? They're only supplying the SA node and the AV node. Can you see the difference between the two innervations? Sympathetic supply is more general. Parasympathetic supply is more on the SA node and the AV node. Now, when sympathetic fibers fire, heart rate will increase, and also the force of contraction, because it's supplying the cardiac muscle as well. When parasympathetic outflow increases, it will decrease the heart rate, but it won't have an effect on force of contraction, because it's only supplying the SA node and the AV node, so it only affects the heart rate. Okay? Make sense? All right, now so parasympathetic fibers release acetylcholine, binds to muscarinic receptors, and sympathetic nerves release norepinephrine or noradrenaline, which binds to beta 1 or beta 2 receptors, beta receptors basically. Now, binding to beta receptors will have positive chronotropy and inotropy. So remember these two terms, forget about the other two. Chronotropy relates to heart rate, and inotropy relates to force of contraction. Sympathetic outflow will cause positive inotropy and positive chronotropy. Parasympathetic will cause negative chronotropy, but it doesn't have much effect on inotropy. Does that make sense? Okay. All right, now let's look at stroke volume now. Stroke volume is the amount of blood that leaves ventricle with each contraction. Yeah. What will determine stroke volume? Preload, afterload, and exercise. We'll talk about it later. Preload and afterload are two major factors. Preload is how well you fill the ventricle before it contracts. If you fill it more, you can have more output, right? So if you increase the filling of ventricle, remember the end diastolic volume? How much was it? 120. If you make it 130, for example, so you increase the preload, you fill the ventricle better, there'll be more stroke volume, right? So increase preload, so if venous return increases, it will increase stroke volume. And the mechanism by which it increases is called Frank-Starling law. Has anyone heard of this before? What is this law? Anyone? What is Frank-Starling mechanism? Anyone? It says if you stretch the myocardial fiber, okay, if you stretch the fiber more, it will contract with more force. So when you increase venous return, the ventricle fills more with blood. There'll be more stretch on the myocardial cells. So when you stretch them more, they will force with more. They will contract with more stronger force. This is Frank Starling law. Okay. So if you fill the ventricle more, it will contract with more force. If you increase preload, the stroke volume will increase. Afterload. Afterload is now when ventricle is contracting, right? Left ventricle, for example, it is pushing blood into the aorta. Now the aorta is a narrow channel as compared to the ventricle, and it is already filled with blood, and there is some pressure in the aorta. So ventricle has to overcome that resistance in order to pump 80 ml of blood into aorta. So afterload is a kind of barrier, a pressure against which ventricle has to push. So if you increase afterload, stroke volume will decrease. But if you decrease afterload, stroke volume will increase. So if aorta is dilated, for example, there is less pressure in the aorta, stroke volume will increase. Okay? So more preload, more stroke volume, less afterload, more stroke volume. Now exercise, people who do regular exercise, athletes, okay, they end up having higher stroke volume. They end up having higher stroke volume. Because of that, because they have higher stroke volume, they end up having a low resting heart rate. You have to meet the cardiac output, right, which is say 5.6 liters. If you have increased stroke volume, you can live with a low heart rate, meeting the same cardiac output, right? So that's why in athletes, the resting heart rate could be 60 or even less, because their stroke volume is larger than the usual population. So these are the three factors that will determine stroke volume. Make sense? Now, preload, afterload. Preload is how you fill the ventricle, and afterload is, you can imagine it like a clamp, the resistance against which ventricle has to push the blood out. So preload is the volume of blood in ventricles at the end of diastole. What will cause an increase in preload? Hypervolemia. If someone is overloaded with water, so there's too much volume of blood, you will have more return to the heart. Regurgitation of cardiac valves. Sometimes the valves are leaky. So, usually the valve should close completely, but if the valves are leaky, more blood fills into the ventricle. Ventricle becomes stretched. Okay. Also in heart failure, okay, preload could increase because the muscle has become weaker, so it fills more. Afterload, resistance the left ventricle must overcome to circulate blood, increased in hypertension. Hypertension means the pressure in aorta is higher now, so the heart will have to pump more with more force. Vasoconstriction. If you constrict the blood vessels, resistance will increase, so heart will have to pump more with more force. So if afterload increases, it causes an increase in cardiac workload. That is the reason people who have hypertension for many, many years, their hearts undergo hypertrophy. The cardiac muscle becomes hypertrophied because their heart is always working to overcome the resistance. So when that happens over a number of years, we will discuss next week, the ventricle becomes hypertrophied. Initially it is beneficial, but after some time, the heart begins to fail. Okay, so relationship of preload and afterload with the stroke volume. Okay, so increase preload, increase stroke volume, increase afterload, decrease stroke volume. Is that concept clear? Okay. Now this is putting everything in context. Stroke volume and heart rate determine cardiac output. What determines heart rate? Your autonomic nervous system, central nervous system, Your emotional states can alter your heart rate. Neural reflexes, hormones like thyroid hormones, catecholamines, etc. Stroke volume determined by preload, afterload, and contractility. So how strong the muscle contracts. Okay, venous return, end-diastolic volume, aortic pressure, so on and so forth. So all these things will determine the cardiac output at the end. Okay, if any one of these is compromised, another factor will try to compensate initially. Okay, if one of the factors is compromised, other factors will come in to compensate for the lack in cardiac output. So as we will see when we talk about heart failure, when the heart begins to fail, it can't maintain enough cardiac output, heart rate goes up in order to make up for the cardiac output. Okay. All right. Now, as I was mentioning to you a few slides back, cardiac muscle is contracting and relaxing all the time, so it requires a lot of oxygen, right? Although the ventricles and atria are filled with blood, but they can't get oxygen from that blood. It's a thick wall of muscles, right? So it should have its own blood supply. When aorta is leaving the left ventricle, it gives off arterial branches, which are called coronary arteries. These arteries will then travel outside the heart, the myocardium, and send branches into the myocardium. Okay, the coronary arteries, the coronary capillaries will supply the myocardium. Okay. Now, looking at the coronary circulation, so this is very important because, as you can see here, number one cause of disease, cause of death, according to WHO, is coronary artery disease. Okay, so number one cause of death around the globe is due to coronary artery disease, also called ischemic heart disease. We'll talk about it in more detail next week, but I'll just give you some idea about the physiology of coronary circulation today. Okay. Now, as I told you, when aorta is leaving the left ventricle, it gives off these branches, coronary artery. Okay. So this is left coronary artery. This one is right coronary artery. Then further branching, and it will supply the myocardium. Okay. Now, what regulates blood flow through the coronary circulation, through the coronary arteries? What do you think? So there are three mechanisms that regulate blood flow through the coronary arteries. The first one is very interesting. Second and third is easier to understand. First one is a bit tricky, but also very interesting. Now, when does the heart pump blood? During the phase of systole, right? So during systole, what is happening? The blood is leaving the left ventricle. I'm just talking about left ventricle. Imagine it's happening on both sides. I'm talking about left side only. So left ventricle pumps, okay, blood goes into the aorta during systole. Yeah, when blood is going into the aorta, it will also go into the coronary arteries. And coronary arteries are passing into the myocardium. Would there be good blood flow at this time in the coronary arteries? Yes or no? Just give me one answer. Yes or no? When the heart pumps during systole, yeah, blood is entering the aorta with full force. It will go into the coronary arteries, right? The immediate branches coming out from the aorta. Would there be good blood flow through the myocardium at that time, within the coronary vessels? Say yes or no. No. Okay, your answer is correct. The reason for that is, at that time, the myocardium is contracting, right? So the capillaries which are passing through the myocardium gets compressed. Muscle is contracting, right? Although the blood is coming into coronary arteries, but it will not go into the capillaries supplying the myocardium, because myocardium is contracting. There's pressure on the capillaries. So the myocardium actually gets better blood flow during diastole. When the cardiac muscle relaxes, blood flows through the coronary capillaries and supplies the myocardial cells. Okay, so the pressure changes in the ventricular wall will determine the blood flow. So during diastole, blood will flow through the coronary capillaries and supply the myocardial cells with oxygen. During systole, the flow is hampered because of the pressure of muscles onto the capillaries. Make sense? Second mechanism is neural, autonomic. Yeah. So sympathetic, parasympathetic will alter the blood flow because there are receptors on the smooth muscles which are in the wall of coronary arteries. When sympathetic binds, it causes dilatation of the arteries. Okay, parasympathetic causes constriction of the arteries. Local. Now this is the most important mechanism of regulation blood flow in case of the heart. Okay, local. So myocardial oxygen demand and local metabolites. What happens? Myocardium is, so these muscles are contracting all the time. So they are very busy. When they're very busy, they're burning a lot of oxygen because they need a lot of ATP. When they're burning a lot of oxygen, they're making a lot of CO2, right? And other metabolites. These metabolites, carbon dioxide, decreasing pH, this causes local vasodilatation. Okay, so these factors will cause vasodilatation of the coronary vessels. So blood flow will be good. Okay, so when the heart becomes more active, there'll be more vasodilatation, so more blood will flow. So it can regulate, depending upon how active it is, its own blood flow. Okay, this is known as local mechanism. It is beyond the control of autonomic nervous system. It's controlled by the heart itself. If the muscle is more active, more metabolites, blood vessels will dilate more blood will flow through the coronary vessels. Make sense? Okay. So these two can be altered. This is something which is, you can't do anything about it. In clinical practice, you have what is known as coronary angiography, very common nowadays. You inject a dye and then you take an X-ray. You can see the coronary arteries. If there is any narrowing, for example, in this case, there is a narrowing of this artery, the arrow C, you can see any physical narrowing of the coronary arteries. It can tell you about the anatomical structure of the artery. You can also do Doppler, like echocardiogram with Doppler technology. So you can actually see the blood flow through the chambers of the heart. You can also see blood flow through the coronary blood vessels. Okay, so you can have an idea how efficient coronary circulation is. Okay. Moving on to systemic circulation. Any questions about the heart itself and the coronary circulation before I move on to the systemic circulation? Any questions from online students? Any question about whatever we have covered until now? The structure of the heart, cardiac output, the factors regulating cardiac output, coronary circulation, all good. Okay. So let's move on with systemic circulation now. When we say systemic circulation, it means the blood flowing through arteries, veins, capillaries, and so on. Yeah. Okay. So you have arteries, arterioles, capillaries, venules, and veins. If you look at the structure of artery versus structure of a vein, this is how they would appear. Okay, so the artery would have a very thick wall, a lot of smooth muscle fibers in the wall. The lumen is narrower as compared to a vein. The vein has larger lumen, but thin wall, so thin layer of smooth muscles. Okay, so arteries are also called resistance vessels because of presence of these smooth muscles in the wall. They can alter their diameter. If they become narrow, they will offer more resistance to blood flow. If they dilate, less resistance. Veins are also called capacitance vessels. So they have the capacity. They don't offer resistance to blood flow. They are rather capacitance vessels. So at any given time in a person, almost 60% of the blood is actually present in veins because of their large lumen, the large size. Okay. But what determines the blood pressure, okay, the resistance that vessels offer to flow of blood is determined by arteries and arterioles, not by veins. Make sense? All right. Okay. Now, what happens at the capillaries once the blood reaches capillaries? This is where exchange of gases, metabolites, waste products will take place. Yeah. So arterial end of capillary, oxygen and nutrients, they'll go out, and CO2 waste material would come in, and the capillaries will return the blood into venules and ultimately into the veins. Okay. Now, there is something known as a venous pump. Remember I told you, veins do not have a thick muscle coat in their wall, and the lumen is quite big, and they can't really alter their lumen, right? And at any given time, 60% of blood is within the veins. And when you're standing in an upright posture, your heart is in your chest cavity, right? And you have all these big veins in your abdomen, in the lower limbs, okay? And they cannot really pump, they cannot really contract. So how does the blood return back to the heart? Against the gravity. There is some pressure in the veins. So when the heart pumps, the pressure is generated, which goes into the aorta, then to major arteries, then arterioles. In capillaries, the pressure is very, very low. In veins, even lower. So the pressure is very, very low. Okay. In order for the blood to return to the heart, there are other mechanisms which are required. Okay. One of them is called muscular pump or venous pump. So see, this is a vein, and the skeletal muscles are around the vein, relaxed. When the muscles contract, they compress the vein. Yeah, they compress the vein. And within the veins, you have one-way valves. One-way valves. Once the blood goes up, it can't go down because the valve will close. Okay, so in the large veins in our lower limbs, in the legs, you have this venous pump. Okay, so when you walk, when your muscles contract, they compress on the vein and blood travels upwards. So that's why when you are on, say, traveling on a plane, a long flight, the advice is given to you, after every half an hour or so, get up, walk a bit, and sit down, so that you're, especially for those people who are prone to develop venous stasis, they should move their limbs more often, contract their limb muscles, so the blood flow is good, okay, through the veins. Make sense? Okay. Now, looking at blood pressure, I already mentioned to you that by the time blood reaches the veins, the pressure is very, very low. So if you can see here, in the ventricle, at diastole, the pressure is very low. At systole, the pressure goes up to 120 millimeters of mercury. In the aorta, you have 120 systolic and 80 diastolic, right? So it doesn't go below 80. That's the usual figure. Now tell me, what is the pressure that left ventricle has to generate during systole so that the blood will flow into the aorta? It should be above 80, right? Remember afterload? So left ventricle has to generate a pressure higher than 80, only then blood will flow from left ventricle to aorta. If it's less than 80, blood will not go into aorta because aorta is at 80 millimeters of mercury, right? So this is afterload, which left ventricle has to overcome. Once in aorta, so you have this fluctuation, 120 over 80. As you go down from arteries to arterioles to capillaries, the pressure begins to drop, and by the time you are in veins or venules, it is less than 20. By the time you reach back to the heart in right atrium, it is almost near zero. Yeah? Very, very little pressure. Okay, make sense? All right. Now, what determines the blood pressure? What determines the systemic blood pressure? When we say blood pressure, we refer to the arterial pressure. This is what we measure, right? Clinically, arterial pressure. What determines the pressure? Cardiac output, how much blood the heart is pumping every minute, and how much resistance the blood vessels are offering to this flow. Okay? So if you increase cardiac output, blood pressure will increase. Or if you increase resistance, which means the arteries become narrow, okay, cardiac output is the same, what will happen to blood pressure? It will increase. So these are two factors. Either you can increase cardiac output and the TPR remains the same, for example, blood pressure will increase. Or if cardiac output stays the same, but you increase TPR, blood pressure will increase. So these two factors determine blood pressure. Make sense? Easy? Now, BP varies in a person. 120/80 is a textbook figure. It's not the real figure. So BP varies when you're sitting down, you're standing up, you're lying down, you're running, you get up in the morning. So there are a lot of factors that alter BP, but it remains within the physiological range. Okay. So the factors that determine BP, physiological factors, age is one. So these both pressures gradually rise with age. Systolic more than the diastolic. The rise in BP with age is greater in males than females. There is diurnal variation, so 24 hours variation. It is lowest during sleep and highest in the morning after waking up. It is also increased transiently during physical stress, mental stress, emotional excitement, so on and so forth. So there are a lot of factors that can actually alter blood